esophagus picture in there. So there's at least a couple of those questions that didn't show up. So we will try to sort that out. And hopefully, at least as we go forward, some of those will show up correctly. Our lab this week is a reproductive anatomy, is what we're up to. So if you didn't see from the announcement today, there's our code for that list. All of it will be in reproductive, in our reproductive section here. And all of those anatomical terms. I included a couple of their overview videos, are helpful to go through those structures. We've got a fair amount of histology in there, and I did not have any of the images in there. There was all a couple of uterine images that were thrown in there. I must not have used on there. So that's our list there. Again, there's post lab on there. We will see what becomes of the lab quiz. I'll need to go back and make sure that my images work. Otherwise, we'll have everything from APR sorted out here. Okay. So our last anatomy lab that we have, so next week you'll take the quiz over this, and then we have a few digestive physiology labs to do through McGraw-Hill, some other hopefully helpful labs to understand some of those physiological concepts that we'll be going over here fairly soon. Another note that I did not put in the announcements, but so we have our, in a previous announcement, I had typed note our coming up exams, and I put one of them as April 1st, I think, April 1st and 2nd, which we don't have class on those days, so we won't have the exam at that point. To that question in just a second. I've had a couple of people ask me that. So we'll actually have it that couple days before, so the 30th and the 31st, the Tuesday and Wednesday before our mid-semester recess, so that week before Easter. We're looking at that on the calendar. Last week's lab quiz, I should have finished grading today, so I'm about a third of the way through those, so they should be updated here by the end of the day, I would imagine. It's one of my main things to do, that and start grading, starting grading your exams. Start getting those finished off here too, to try to get done by Wednesday probably for those. So for this exam grading, I was going to start in the middle of the alphabet. So if your name starts with or last name starts with a M or an N, you'll probably get your score here sometime today. And then if you're A or Z, it'll be a couple more days. So I'll work my way out to get those done. Try to get them done as quick as I can. So once those are graded, I'll put them all the stuff available for you to see, all the questions and answers and scoring on those. Questions on that? There's a couple emails and messages I got sent. Making sure I answered those questions. All right. So our reproductive anatomy, there is two different sets of structures to look at here, and a bit of histology that can be a little difficult for some people. This is one of those that we frequently, I don't know if it's because it's closer to the end of the semester that people start not paying as close of attention to it, or if it's genuinely difficult. Oh, so one last question. Apparently there was a picture on the exam too that the picture didn't show up. It was supposed to be an oxyhemoglobin dissociation curve. So that I got a bunch of emails about that. So that question will be thrown out from the exam if you had questions on that. Okay. So for reproductive anatomy, we'll be taking a look at these two sets of internal and external portions here of the reproductive system in males and in females. This cross-sectional view is a very handy one to start with, to compare and contrast what we're looking at. So in both of them, there's a couple of common things to pick out. So you have the pubic symphysis here in front, what we're looking at with the bones. We can see the behind the pubic symphysis, the urinary bladder, and then the back of the image, you can see the rectum. So we have those three. The external anatomy here in front of the pubic symphysis in males and females is an area called the mons pubis, just a fat pad that sits on top of the pubic symphysis, and that'll be above the external genitalia, so the penis and the scrotum in males, and collectively the vulva in females. That'll be the front there. Behind, again, the rectum will open to the anal canal, out to the anus. So in between there will be the external genitalia, and then we've got a reproductive tract, so a ductal system here that in the case of the males is going to start in the scrotum, the testes, the epididymis, and the vas deferens, also known as the ductus deferens, so either one of those. So the epididymis has a head, a body, and a tail. So these are all convoluted tubules. This is going to be where sperm production is going to take place. It's going to pass through this twisting epididymis, and then the vas deferens, or the ductus deferens, is going to go up a structure called the spermatic cord and wraps around to the back of the urinary bladder, where it's going to be joined. So that's going to be a paired system. There's two testes on each side, and those will be paired with two seminal vesicles, which is going to, when those join together, the seminal vesicles, the duct of the seminal vesicles, and the vas deferens are going to join into this ejaculatory duct, which is going to run into the urethra. The prostatic urethra is going to be the path out from the urinary bladder, and in the male reproductive system, that's going to be combined with urinary tract rather than being next to it like in the female tract. That ejaculatory duct will join here in this round organ below the urinary bladder called the prostate gland, and then that's going to pass through the internal and external urethral sphincters are going to be below that. And we'll see a bulbourethral or Cowper's gland that also has secretions in here. So we'll look at these organs, the seminal vesicles, the prostate gland, and the bulbourethral gland are all going to be organs that contribute to the secretion known as the semen. Right? So our path through there will then go into the spongy urethra here. So again, a duct carrying out of the body. So the urine is going to pass through here, and also the secretions here, the seminal secretions from the testes, so the spermatozoa, and then the seminal vesicle secretions, prostate and Cowper gland secretions will pass through there. Through the portions here of the penis, we'll look at the corpus spongiosum, which is the darker purple here, and then the corpus cavernosa, or corpora cavernosa, which are going to be these structures above when we look at the structures there of the penis. So our external genitalia, penis and the scrotum, contain the testes that'll be comparable to the ovaries in females. This will be our sites of sperm and egg production, and then that'll be attached to a tubular system, a ductal system, that'll carry that out of the body. In the case of the male reproductive tract, that'll join here with the urethra. So those two systems are going to start very similar to one another. The testes and the ovaries are both going to be here in the pelvic cavity. So just above that, you can see the peritoneal membrane would be just above that. Pelvic cavity will have the testes and ovaries here in these same locations, in the ductus deferens, the vas deferens in males, and in the females, that'll be the fallopian tubes. Will run down and eventually out of the body. But you can see during development, you'll begin to see development in males of the scrotum. You can see this long tendon, gubernaculum, that's going to be attached to the testes. As the scrotum descends, that's going to pull that down here along with a portion of the pelvic cavity. So as that gets pulled down, the pelvic cavity is going to form this inguinal canal. This is going to be a path that the vas deferens, the blood vessels and nerves for the testes are going to pass through here, through this inguinal canal as well. So this is an opening in the muscular layer here. Again, this is another common site for males to get hernias. We mentioned the hiatal hernia is common in both this inguinal area, this inguinal canal, is a common site for a portion of the intestines, for example, can get pushed through there if you get really high pressures above that. So as that descends, again, a portion of the pelvic cavity that's going to have a visceral and parietal layer on it here is going to get pulled down with the testes here into the scrotum. The vas deferens is going to get pulled down through that inguinal canal and form the structure called the spermatic cord. That projection here of the pelvic cavity is going to get closed off and have this layer around a portion of the testes called the tunica vaginalis. That tunica vaginalis is going to have this double layered portion around at the edge of the testes here. We'll just have, excuse me, a double serous membrane in there. It'll arise from that same membranes here that make up the pelvic cavity. So you can see the portions here, again, the vas deferens. This is what a normal descending testes will look like. That's something that can be a problem during development. Then it may take longer for a portion of the testes to descend, or you can have a portion of the vas deferens that doesn't descend correctly. So there's all kind of variations that can occur here during development. Others include maintenance that the tunica vaginalis may be larger or smaller. So you can see variations in the size of how much of that cavity gets pulled down there. Right, so they're going to get pulled down. Here's our external view of the genitalia, showing the perineum. So the perineum is this area, comprises the entirety of this area, showing our anal and urogenital triangles. So both of those together are called the perineum. So an important distinction to make there between the two of those, they're collectively called the perineum, not the peritoneum. So the peritoneum is in our abdominal cavity. The perineum refers to this area in both male and female external genitalia and anus, so the region in between there. So again, that mons pubis, just above the pubic symphysis, is the front of that. The coccyx, our tailbone, is going to be the back. And then our ischial tuberosities, think of the projections from the bottom of the ischium, that backbone of the pelvis, two bones you're sitting on, form the outer portions of that. So the perineum has these two portions, the urogenital and the anal triangle between the two of them. So a couple anatomical distinctions to make. And to go back to that, when we look at the external genitalia, is our best view we have here of the scrotum. There's two testes on either side, tunica vaginalis. At one side of those, in the epididymis, that'll run into the vas deferens on the back sides of those. Those are going to be separated by what's called the raphe. And then there's a thin muscle around the, just beneath the skin here, called the dartos muscle. And the dartos muscle is going to be able to contract and relax, and that will cause the testes to descend or get pulled closer to the body. So sperm production is ideally a little bit below body temperature here. So raising and lowering of the testes is going to accomplish that depending on external body temperature. The penis here, the structure not shown here at the edge here is the foreskin or the prepuce. We're just looking at the very edge. There's a small flap of skin that folds back. A male circumcision involves removal of that small bit of tissue there, which is fairly common in the US, and then in Jewish culture that's common. Most of the rest of the world is not very common. So primarily cultural reasons that you'll see that removed. There's a tiny bit of evidence, not strongly supported, that male circumcision might decrease transmission of some sexually transmitted infections, but not strongly supported. So it's just something that people have done for a long time, and so that's the way things have just remained as a result. But there's just a little bit of nervous tissue in there, a little bit of vasculature. We'll talk about that compared to sometimes referred to as female circumcision is a very different process and is more accurately called genital mutilation, if you've ever heard either of those. So very different in comparison between the two. So taking a look at those testes, again, you can see the testes, the ductus deferens, the epididymis around there, the head, the body or corpus, and the tail becomes the vas deferens. You can see that arising. If you look on the right side of the image, you can see the dartos muscle surrounding that can contract and relax the and pull the testes closer or further as it relaxes, move them close further away from the body. The vas deferens here, you can see going up this structure called the spermatic cord that runs through the inguinal canal. So in addition to the ductus deferens or vas deferens, blood vessels and nerves that supply the testes go through there. So remember our testicular arteries and veins, our testicular nerves, we just referred to those as gonadal back then. So testicular going down to the testes here. And those are surrounded by a muscle called the cremaster muscle. I thought was labeled on here. Yep, there it is. So this circular muscle that surrounds the spermatic cord that can likewise, when it contracts, will pull the testes closer up to the body. When it relaxes, it will lower the testes further down there. So that passes through this inguinal canal. The vas deferens, again, goes to the back of the urinary bladder and joins with the seminal vesicles, again, which are going to be paired. So there's two vas deferens and two seminal vesicles. They look a little bit like little wings at the back of the urinary bladder. If you've ever dissected done any dissections in other classes, like a rat or a pig, these kind of stick out at the back of the urinary bladder here. So seminal vesicles, again, the ejaculatory duct will join with our prostatic urethra and pass through the prostate gland here and into the spongy urethra and out through the opening of the urethra there, the orifice where that'll pass through. So after the prostate, we have this, the urethra is then going to pass through this structure called the corpus spongiosum. And that structure is going to be bounded on either side by these dual structures called the corpora cavernosa. These two structures, all of those are going to be these very highly vascularized structures that can have a lot of blood flow through them. We'll see those in a little bit more detail in the next couple of slides. But those are going to be, the corpora cavernosa are going to be the structures that fill up during an erection here in the penis. So we'll see that primarily regulated by nitric oxide. Talk about that process a little bit. As we learn about that process a little bit, the discovery of nitric oxide as the molecule that caused smooth muscle relaxation was the driving force with that discovery led to the discovery of a number of drugs that influence that, including all the things that you'll see during a baseball game or a football game, like Cialis and all those erectile dysfunction drugs. So if you want to point to the primary driver of a lot of research in biotech companies, it's not necessarily what we can do to best help other people, but what we can do to make a lot of money between those. So that was one of the first big drug discoveries that came out of nitric oxide discovery. So our corpora cavernosa and corpus spongiosum, the urethra is the spongy urethra passes through the corpus spongiosum. Then we go out to, those are within the penis. The glans penis is here at the end. We'll see those structures here on our next view here. The glans is going to be surrounded by that flap of skin called the prepuce, the foreskin around there. So a couple parts of the cavernosa here has the crust here at the end, has that the proximal end here of the corpora cavernosa, and there's a couple of muscles here, skeletal muscles that are at the base here, the root of the penis, around the bulb. There's a number of skeletal muscles that are present there, and this is where we have the external urinary sphincter right through there, just in front of the prostate. And the prostate is an organ that during the course of a man's life gradually increases in size. So measuring, there's a test that is not very accurate for prostate cancer called the prostate specific antigen. So there's a protein that's made in the prostate. And as the prostate gets larger and larger, it's going to make more of that protein. In males, as the prostate enlarges, that PSA or prostate-specific antigen will increase. So it tends to result in a lot of false positive tests, though, and it is not necessarily a very successful marker for prostate cancer. But it does indicate an enlargement of the prostate. And as that prostate gets larger, it starts to push up against the urinary bladder and can limit flow through the prostatic urethra as well. So decreased flow rates through there, problems with urination become a major problem in both males and females, as we'll see for both of them. But that's the problem that arises in males. This is one that using the rough estimate of 50% of males over 50, 60% of males over 60, 70% of males over 70 have problems with an enlarged prostate. So very common part of aging. In the cross section here of the cavernosa and the spongiosum, again, cavernosa are paired to either side there, and then the spongiosum surrounds the urethra. A couple views of those two. Through our, going back to the testes, and this will lead into some of our anatomy here, or our histology rather, is taking a look at the structures of the testes. So again, paired organs, this is going to be our site of sperm production. And those are going to start here in the seminiferous tubules. They're split off into these lobules, so these coiled tubules that are going to be split by this connective tissue that forms these different septa. The individual septum is the empty space that the lobule is sitting in, that the seminiferous tubule is sitting within. When we're looking at cross sections of the testes, the primary structure that we're looking at are these seminiferous tubules. We're looking at sections of these convoluted tubules, these twisting tubules that are going to be the site of sperm production. Those seminiferous tubules are going to flow through what are called the tubulus rectus and the reedy testes here. All those tubules will run together into the efferent ductules that go into the head of the epididymis. Those will be joined together in the head of the epididymis into a single tubule that'll be coiled around the body of the epididymis and into the tail of the epididymis. So there'll be a fair amount of storage that occurs here. So during production of the spermatozoa that are going to be here in the seminiferous tubules, they gradually will move and be stored in the epididymis. And then during ejaculation, there will be contraction of the seminiferous tubules and also the epididymis to push things into the vas deferens. So spermatozoa will be stored in there. If they don't get moved out via ejaculation, will just get absorbed back into the body. Those will pass through there. So through the tail of the epididymis into the ductus deferens or vas deferens, it'll run up that spermatic cord. And our membrane that surrounds the testes, so the tunica vaginalis will be outside of that. The membrane around the testes itself is a membrane called the tunica albuginea. That will be the membrane around that. The vaginalis will be hopefully right out here. Membrane sitting right outside of that. The albuginea will be this additional membrane that we have here. We'll talk about, this will be another place where we have a vascular barrier as well, a blood testes and blood ovarian barrier that'll keep things a little separate here, or an additional filter at the very least. So when we look at these cross sections of the testes at the seminiferous tubules, looking at a very close view here, so on the upper right here is a cross section of that tubule. You can see the outside of it here, and the lumen, it's a hollow tubule that's passing through there. We'll have our germ cells, the spermatogonia, the plural of that, that'll be undergoing mitosis and meiosis to get us our, eventually our sperm cells. So we'll go through the process of, you've had this a few times in different biology classes, that process of taking our 46 or 23 pairs of chromosomes and splitting that into our sperm cells that are going to just have a single pair of 23 chromosomes in each one. It'll fuse with our ovum, our egg cell, upon fertilization. So we'll have those spermatogonia, and as we go through mitosis and then through meiosis, we'll also see eventually the development of these other processes in the cell. So the spermatozoa, in addition to undergoing the split, is going to develop a number of specialized structures that are going to be present in the spermatozoa or the sperm cells. We'll go through our primary and secondary spermatocytes out to the spermatids, and then the spermatids will become spermatozoa and detach in the lumen and get pushed through there. So one of the structures here that's notable on the spermatozoa is going to be the presence of a flagellum. So the only cell that has a flagellum in the body, they are not going to be mobile. Those are not going to be motile. They're not going to be moving until ejaculation. So as they pass through the seminiferous tubules and into the epididymis, it's just going to be the same type of movement that we've seen in tubes anywhere else. It's just going to be pressure differences. So contraction here and production of other cells will create a pressure difference to move those cells through the seminiferous tubules. Our division here, as we undergo the first division, the spermatogonia divide, they're going to pass through our tight junctions on these cells that have a couple of different names here, labeled here as sustentacular or Sertoli cells, also called nurse cells. These are going to be a supportive cell, and also they're going to form the blood-testes barrier. So once the cells divide and move across and become primary spermatocytes, there's going to be an additional filter that's present on here for the blood to flow through. So that anything coming through there, in particular, immune cells are not going to be able to make their way through there. So this provides a barrier because these cells are now not going to be expressing similar surface proteins compared to the other cells in our body. So they would be recognized as foreign cells because they're not going to have the same complement of DNA that we have throughout. Right? We're going to be left with just half of our initial DNA from the germ cell. So as we pass through there, those Sertoli cells or nurse cells or sustentacular cells will push things into the inner membrane here. So as we move forward, primary and secondary spermatocytes are going to be fairly similar in location, and then the spermatids are going to be projecting out into the lumen, and the spermatozoa or sperm cells will be in the lumen here of the seminiferous tubules themselves. So on the left, that similar drawing that what we saw, again, are germ cells, or spermatogonia here, the primary and secondary spermatocytes, the spermatids, and then the spermatozoa within the lumen here of the tubule, of the seminiferous tubule. Some of the surrounding cells here we can see as well called our interstitial or Leydig cells. These are the testosterone producing cells of the primary testosterone producing cells in males. Those will be around the outside of the seminiferous tubules. We're looking at a seminiferous tubule within the testes. If you had a picture like this and asked you to name the organ would be the testes. The tubule that we're looking at would be the seminiferous tubule. Here's a closer up view of that. A couple things labeled here are spermatogonia, labeled SA and SB here. These are the germ cells. You can see a couple of these sustentacular cells in between there, again, forming those blood-brain barriers. You can see our primary and secondary spermatocytes as we're going through after mitosis and going through meiosis. Do these cells in the intermediate layers here. And then you notice in the formation of the spermatids, the nucleus now has a much more oblong appearance, of a bullet shape that you'll see to spermatid nucleus compared to the rounded nuclei that we have elsewhere. And then around the edges, not a lot of separation. So we have our Leydig cells or interstitial cells around the outside of the seminiferous tubes. Here's another view of that. So again, the Leydig cells are interstitial cells in between here. And then within the lumen, a couple different numbered lumen that we have in here. You can see at the surface those kind of thinner nuclei for the spermatids that we see at the surface. Our in-between cells are the primary and secondary. You can see some of the spermatogonia around the sides, and then some of these sustentacular cells that are in between. Notice the clear membrane, the identifiable membrane of those sustentacular cells. SC are spermatocytes, primary and secondary spermatocytes. You see a few of these sustentacular cells that'll be at that bottom layer as well. A couple of the spermatogonia, again, right at the outside is what we're looking at with those. So here we're looking at the testes. Those testes are going to pass into the epididymis as our other section here of the male tract to look at. So the epididymis, again, that next set of structures is going to be characterized by these very tall ciliated cells. You can see the small lines you see projecting in there. Those are pseudostratified columnar epithelia that we have with very long cilia. These cilia through the epididymis are going to be moving our spermatozoa, which are here in the center, through the epididymis. And there's a ring of smooth muscle here around the outside as well that can contract and push those through the epididymis. So during ejaculation, you have contraction here of the epididymis that's going to move things through the epididymis and into the vas deferens. So our other, that's our few slides there of our male anatomy, histology there. And then our other section is the female anatomy. So going back to that same picture, the similar picture that we saw in the males. Again, we can see the pubic symphysis with the mons pubis in front of it, the urinary bladder sitting behind it, and then at the back, the rectum into anal canal. And wedged in between that is going to be the female reproductive system, which will be above the urinary bladder and the pelvic cavity rather than below it and externally that we see in the males. So behind the urinary bladder, you can see the uterus, and the uterus is composed of smooth muscle and a mucosa here. So this is the thickest smooth muscle in the female body, referred to as the myometrium. sections there. The uterus are the myometrium, is our smooth muscle portion. And then we have the endometrium, is the mucosa in there. Above that, we can see the uterus. You can see a round ligament that runs from the ovary to the uterus, and then our fallopian or uterine tube that runs out and is actually open to that. So the ends of the fallopian tube have these little finger-like processes called the fimbriae. Those fimbriae sit near the ovary, and upon ovulation, when the egg is released from the ovary, those fimbriae are going to help bring that into the fallopian or the uterine tube. That egg can eventually pass into the endometrium if we're looking at implantation. So fertilization will usually occur in the tube itself when we're looking at the joining of the sperm and the egg will occur out here in the fallopian tube. And then that fertilized egg will get implanted into the endometrium here in the uterus. Those structures there. We'll see a couple views here of the uterus. It's going to tail off. It's going to narrow at its base here. So we have our cervical, our neck portion of that. So the cervix of the uterus then opens up into the orifice below that, which is the vagina, which will open onto the vaginal orifice, and the entirety of the external genitalia of females is called the vulva. Which will be composed of the clitoris and its portions, opening of the urethra and the vaginal orifice, and the labia majora and minora, or you'll see those alternately called the labia minor, the labia major, or APR labels them as the minus and the majus. So those structures of external genitalia. An older term that we still see some remnants of here, the word pudendum or pudenda was a term initially used for this in Western culture. So the Latin word pudendum, I think I mentioned maybe in our vascular, probably in vascular, maybe our neural sections. So the arteries and veins supplying this area are still called the pudendal artery and the pudendal vein. But the word pudendum means shameful. So at the very least, the external genitalia, you'll much more often see the word vulva used there, which doesn't have the negative connotations and the hatred of women's bodies implied by calling it a shameful part, because the word vulva just means a covering. So our similar structure when we saw the external genitalia, again, the perineum here would run in roughly the same line. Our urogenital and anal triangles there to the mons pubis. So the vaginal orifice, urethral orifice above that. Again, you see that term pudendal cleft or cleft, referring to that opening vaginal orifice and the urethra above that. The labia minora and labia majora surrounding that. At the front of that, we have the prepuce, so similar to the prepuce in on the surrounding the glans penis in males. There's the thin flap of tissue there, also called the clitoral hood, and that'll surround the clitoris. It actually runs down a little bit to the sides here and has a few different sections of it here. But within the clitoris, we have a comparable structure to the corpus or the corpora cavernosa. So during arousal in females, the clitoris likewise fills up with blood and becomes erect in the structures surrounding that. So those external portions here of the female reproductive system down there. Here's the internal anatomy, a lot more to see inside of there. So again, the vagina will be continuous with the uterus. So the cervix, again, is the opening there. So the cervical orifice will go into the uterine cavity. The uterine cavity is going to be lined with the endometrium. The smooth muscle around that is the myometrium. There's a serous membrane around the uterus, sometimes referred to as the perimetrium, or just serous membrane around that. We have two notable ligaments here, the broad ligament, which extends out and holds the fallopian tubes largely in place, and then the suspensory ligament that's going to attach to the ovary and hold that in place. Those two running through there. The fallopian tube or the uterine tube, you can see the opening here. There's a narrow isthmus, this narrow portion before it flares wider. And our ampulla here, the wider portion, again, is going to end in these finger-like processes called the fimbriae. So fimbria is the singular, fimbriae, the plural of that. So the way those move is kind of interesting. It's just moving as the heart is beating. So we'll see a video of ovulation occurring in an ovary, and as the heart's beating, that's going to cause changes in pressure in the pelvic cavity. And so that's going to cause the fimbriae basically to move in time with the beating of the heart. So we'll see these fimbriae, these little finger-like processes that aren't moving because they have smooth muscle in them or anything. They're just moving because the heart contracting is causing this wave moving through the body. And so the fimbriae are going to move over the top of the ovary, and when ovulation occurs, when the eggs release from the ovary, it'll usually pick up the egg and bring it here into the fallopian tube. Pressure will cause that to flow through there. So our couple of, sorry, I didn't see that question there. Oh, that's a while ago. Is the cremaster muscle smooth or skeletal? It is a skeletal muscle, but it does not have a lot of primarily reflexively controlled. Forming that around that spermatic cord. Okay, so here's a drawing showing a cross section of the ovary. So the ovary is going to be the site similar to, comparable to the testes in males and females. This is going to be the site of ovum production. So similar to that, we'll have these structures that are going to undergo cell division. There won't be any mitosis in occurring any longer in here. We're just going to have meiosis. We're just going to be splitting and maturing these cells. From our oogonia. So we're not going to have additional daughter cells formed here. There's only a set number of the oogonia, of the germ cells. That's not going to be the case in males. So it's a difference we'll see in terms of the number of egg cells is going to be limited in the women's body. In males, the number of sperm cells is going to be have a much higher limit because the spermatogonia are going to be dividing and making daughter cells in the process of spermatogenesis and oogenesis in the formation of our ova or egg cells. Those daughter cells, there's not going to be those daughter cells of the germ cell of the oogonia. We'll have this process of our primordial follicles becoming primary follicles, and that'll be largely determined by, in terms of our histological appearance, by the formation of a line of cells here making up the structure called the follicle. So the oogonia, the cell here within the primary follicle, will begin to develop, and that'll stimulate the proliferation of what are called the follicular cells. These will start to enlarge, and eventually you'll start to see a few layers of those follicles and the formation of a fluid-filled chamber around the egg cell as well. So this follicle will continue to enlarge. You can see our secondary and our mature, aka Graafian follicle. So this follicle is going to continue to get larger and larger and larger until ovulation, when the follicular cells and this ring of cells around it, the cumulus mass, will get ejected out of the ovary here, and the remaining parts of the follicle will collapse and form what's called the corpus luteum, literally the yellow body. That corpus luteum, compared to the follicle, we'll see a couple of hormones later on that we've mentioned in 321, luteinizing hormone and follicle stimulating hormone. You can guess at least the basics of what those, well, the name's right there for follicle stimulating hormone. So those FSH and LH are going to be stimulating, obviously, growth here of these structures and maintenance of those structures, but we'll also see them stimulating the secretion of estrogen and progesterone. We'll go through that in quite a bit of detail here. Our outer portion of the ovary, just like the testes, will have the tunica albuginea, and then an outer membrane around that, a serous membrane surrounding that. Looking at a more internal organ. We'll have this selection of primordial follicles, and as the follicle proliferates, we'll begin to have this notable structure, the follicular cells, as they proliferate, are going to fill in, form what are called those granulosa cells. We'll have a layer of fluid that surrounds it as we look at the primary follicles. That zona pellucida will remain around our oocyte here, our egg cell. And as we go from a primary follicle to a secondary follicle, we'll begin to have this separation of multiple layers here that'll eventually lead to the formation again of this antrum and the cumulus mass. It's going to surround, maintain its structure around the primary oocyte here. So upon ovulation, we'll look at the changes within the cell that's going to convert that from a primary to a secondary oocyte upon ovulation. So we've got our oocyte, primary oocyte here as we have a selected follicle, and then upon ovulation, our secondary oocyte. And then the formation of our corpus luteum from that remaining mass. That eventually degrades and forms what's called the corpus albicans. So again, we'll see this buildup of a fairly large supporting structure around this. It's going to aid the development of the egg cell, of the oocyte, leading up to its release upon ovulation. And the corpus luteum is going to continue to support indirectly the continued development of that cell if it gets fertilized. If it gets fertilized by a spermatozoa, the secretions here from the corpus luteum continue to aid in its development, the hormones that are going to come out of there. Here's some images showing what we're looking at here. We can see some primordial follicles, these very small structures, fair amount of connective tissue that we've got here in the and vasculature in the ovaries as well. And then when we look at our primary follicles, like this one here, again, you can see a few layers of those granulosa cells, follicular cells as they're proliferating around there. And then our primary oocyte, we've got here in both of these. So from there to there, we're not looking at much of a difference here between our primary and secondary follicle. You can see the zona pellucidum starts to become a little bit more pronounced, and we would usually start to see a little more space showing up in the secondary follicle here. A little bit more fluid that starts to accumulate inside of that. These granulosa cells are going to be the primary estrogen secreting cells here in the body. So more typical of what we'll see in a secondary follicle, we'll start to see the development of the antrum and the cumulus mass, aka the cumulosa ooporus is another name for that. This mass of cells that is forming around there. FA is our follicular antrum. Antrum, the space in here. And our granulosa, zona granulosa is the region around that. So we're getting this fluid accumulating in here. What's happening during development? So during the course of an ovarian cycle in a woman's body, a number of these primary follicles begin to develop, primordial into primary follicles. From that, one will typically be selected to continue its development. The rest of them degrade. So while there's going to be millions of follicles in the ovaries, only a scant number of those are going to develop into mature follicles and produce ovulation. So between the onset of the uterine cycle during puberty and then ending in menopause, right? You're looking at roughly, give or take a few years, about 40 years. So 40 times 12, 480, 400 to 500 ovulations in that time period. And there's going to be a few million primordial follicles that are going to be present. Only a portion of those develop. So we'll look at that process of selection when we look at the physiology in here. So our secondary follicle, again, zona granulosa, the outer portions of this have our two portions of what's called our theca. So a theca interna and a theca externa. The term we're looking at for those two. So not a huge distinction between those. We usually see the theca and interna and externa have a little clear separation in color, and we can see it in a few of our subsequent images here in the mature follicle. Not a great view if you're struggling to see the difference. It's not a crazy obvious view to see the difference between the theca there. But you should be able to start to see that here. So the theca interna right here, just outside the epithelia here of the zona granulosa, this darker stained ring, and then the much thicker layer of the theca interna is what we're looking at here. Here's the interna, roughly right there, and the externa right there. Right, so this is looking at a mature, also known as a Graafian follicle. So secondary oocyte, you can see that zona pellucida or zona pellucidum. Surrounding it is that clear ring of fluid around it. The corona radiata is the layer of cells surrounding that. The zona glomerulosa are the layer of cells making up the outer portion of the follicle. And then there's a large fluid-filled chamber called the follicular antrum. And you can see all of these simultaneously if you look at a slide from an ovary. So on the edge here, you can see portions of a mature antrum over here. Right, here's the follicular antrum right there. Here's the zona glomerulosa. You can see a portion of our theca interna and theca externa here. And then at the outer portions, there's a number of primordial follicles, are primarily what we're looking at over here. And then a few primary follicles right there. We'll show that a little closer on our next slide. Primary with just a couple layers there of our granulosa cells, these follicular cells that are proliferating around the egg cell, the oocyte present in there. Primordial follicles will not have that larger cytosol that we'll see present in the primary follicle, as that cell is going to be undergoing a lot more activity and thus quite a bit larger. Those primary follicles versus primordial follicles. And then as we progress to our secondary follicles here, again, the zona pellucidum becomes very prominent, and we'll start to see a follicular antrum start to develop here in those secondary follicles. And then the mature follicles, again, a large follicular antrum, that large corona, and the quite enlarged primary oocyte that we see around that. Theca starts to become pretty prominent at that point. So there are a couple images there of the ovary. Our other histology slides that we have here for female is our is uterus. Yep. And we can see the difference between the uterus here in terms of our endometrium up here above and our myometrium below. Okay. So the endometrium is going to have these couple of fairly prominent two glands that are going to develop, these tubular glands that develop in the endometrium. This is during what's called the proliferative phase of the endometrium, where this layer is going to be getting thicker. So the uterine cycle and the menstrual or uterine cycle are going to be overlapping with one another and are controlled by the same processes. So uterine function is going to determine, ovarian function is going to determine uterine function. So during each cycle here through menses, through the menstrual cycle, the majority of the endometrium is going to get built, going to get thicker and thicker during the proliferative phase and begin to develop these endometrial glands, which you can start to see here within the endometrium, that are going to produce a very nutrient-rich secretion from there. So onto the surface, because those nutrients are going to be needed if there is a fertilized egg that gets implanted in there. It's going to need a lot of nutrients to continue to divide and grow during fetal development. So you see this proliferative phase as these layers get thicker and thicker. You can start to see that proliferation. So we've now got a much thicker layer here. Just the bottom here showing our, actually a little higher, sorry, our myometrium separating. So everything above this is endometrium. You can see these glands here are starting to get fairly twisty, starting to get fairly convoluted, but they're not in super high density. We don't see a huge number of these large endometrial glands yet. And that'll be typical of what we see during the proliferative phase, is what we're looking at here. So both of those, this image and the previous one, would be more typical of what we'd see during the proliferative phase. So the endometrium is starting to get thicker and thicker and add more cells. And then as we get to after ovulation, we'll move into the secretory phase. So the secretory phase is going to be notable for a continued size. So we can see the bottom of the endometrium here, just in the bottom left. So everything above here is endometrium, myometrium below it. The myometrium is not changing in size, but the endometrium has gotten quite a bit larger. And these are these very large twisting glands that we're looking at throughout. So very high density, these glands are developing and getting thicker and larger throughout the secretory phase. So their secretions are becoming quite a bit more prominent and more viscous, meaning thicker throughout this process too. Those secretions here start to become very prominent here during the secretory phase. And we'll see these very large endometrial glands. Here's a view showing the comparison between those. So early proliferative phase here on the left, getting towards the end of proliferative phase here in the middle, and then secretory phase on the right. So this cycles back. So the drawn tool there again. So the majority of the endometrium will get sloughed off during menstruation, during menses. And that will regrow again during the next cycle. So you have this huge amount of energy that gets expended to make this layer roughly once a month, right? 28-ish days for that cycle to go around. So it's a huge amount of expense for that to occur, right? And we see that in answered for in the female body in a lot of different ways. So additional fat deposits. So females will have quite a bit more fat deposits on their body because they need to have energy for this process to continue. And the way that they store fat, when you look at the biochemistry, the reactions that drive things like lipolysis, the breakdown of fats, things happen a little differently in female bodies that they tend to hold on to fat quite a bit easier. So what that points to is problems more so typically when you look at some of the applications of that. Females tend to have more trouble with things like weight loss because their bodies hold on to fat quite a bit easier. And under life-threatening situations, females tend to do quite a bit better because, well, probably multiple reasons, but one is their ability to survive under more difficult conditions than males typically. That ability to store fat ends up being somewhat of a double-edged sword. Hopefully, we're not having too many people testing the starvation end of those things too often, though. Right. So a view of our endometrium is the primary thing we're looking at. Again, our myometrium is going to be below that. So our uterine sections there, our phases of the uterus need to know, and then our ovarian sections and the different structures in those follicles are the kinds of questions you'll have here for our next quiz and on your post-lab assignment. Any questions on any of that? Missed anything in the chat?
I guess you were talking about earlier how on some of these slides they have different names than what we use. Like I know I use the anatomy thing to study. Those post quizzes are amazing to use to study. So if they have different words, do you want us to put the ones on there or yours?
I would accept either one of those. So the one I mentioned, like the labia minora versus the labia minus versus the labia minor. Those are all the same thing. So I'll take any of those answers in that case. So just like Craftian follicle, mature follicle are the same thing. I can't remember what APR has it labeled as. I think mature follicle. Other questions, concerns, comments?
I have a question about the exam that we just took. I had one of my written responses, like it skipped on me, so I wasn't able to fill it in. So is there anything, do you have any suggestions what I should do?
Is this the, I apologize, let me talk to you afterwards here, because I think you sent me an email and I misinterpreted what you said in there.
Okay.
We have a few minutes. Other questions? All right. Have a nice week then. No lecture quiz this week, if you noticed that. So a little week off from that, but we do have our homework assignment. And there's a lot of information in lecture this week. We have not started any of those.